shedding and the subsequent loss of the protein coat, vesicles are targeted to specific acceptor compartments. This could either be another organelle or plasma membrane via the process of vesicle targeting, into which they unload their cargo upon membrane fusion. This is a multi-stage event. The first step is recognition of the target compartment following an interaction called tethering. The subsequent interaction brings the opposing membranes much closer to each other and is called docking. Docking leads to membrane fusion. Tethering occurs after the movement of the vesicle towards its specific target compartment. This movement through the cytoplasm to its desired target is largely mediated by diffusion and microtubules with their associated motor proteins. It is tethering proteins which mediate the initial contact between the vesicle and target membrane. Tethering proteins themselves are mediated by Rab-GTPases, which cycle between an active GTP-bound state and an inactive GDP-bound state. When GTP activated, Rab-GTPases recruit Rab effector proteins. Some effector proteins will form a link between the vesicles and motor proteins to aid in the movement towards their target, and others will act as tethering factors to assist in docking the vesicle to its membrane. Despite the efforts of tethering proteins to draw the vesicle and target membrane into close association, fusion itself is energetically unfavourable. To ensure fusion occurs, key proteins called snares interact on either side of the two membranes. There are two important members of the snare family: vesicle snares, known as V snares, and target snares, known as T snares. V snares become incorporated into the membranes of transport vesicles during budding, and T snares are located in the membranes of target compartments. An example of snare mechanism includes the docking of synaptic vesicles during neurotransmission. Snares, syntaxin, SNAP25, and synaptobrevin are responsible for ensuring the synaptic vesicles dock in order to release neurotransmitters into the cleft. Both syntaxin and SNAP25 act as T snares, whereas synaptobrevin is located in the vesicle membrane and acts as a V snare. The formation of the trans-snare complex is achieved by the contribution of one alpha helix from both syntaxin and synaptobrevin and two alpha helices from SNAP25, thus forming a 4-helix bundle. This formation is aided by the binding of syntaxin to the protein MUNC18, consequently increasing the stability of syntaxin within the complex. TRAPs, transport protein particles, are important proteins involved in the tethering process. A TRAP complex on the target membrane recruits a GTPase of the Rab family. The tethering protein captures the transport vesicle by linking it to the TRAP Rab-GTP complex. Binding causes the protein that blocks T-snare binding to be released from the T-snare, which is then free to bind to the V-snare on the vesicle. As the membranes approach each other, V and T snares from opposing membranes interact to form 4-helix bundles, known as a trans-snare complex, and docking occurs. Each bundle contains alpha helices which zip together to form a tightly woven complex that pulls the two opposing lipid bilayers into close association. The hydrophilic heads in the bilayers rotate, causing the membranes to merge, which is known as hemifusion. In this state, the membranes are chemically unstable, so complete fusion subsequently occurs, allowing vesicle cargo to be transferred to the acceptor compartment. Once the vesicle has fused with its target membrane, Rab proteins hydrolyze their bound GTP and the inactive Rab-GDP protein returns to the cytoplasm. The trans-snare complex is also hydrolyzed so that T and V snares can be recycled. This dissociation is mediated by the adapter protein alpha SNAP and the ATPase NSF present in snare proteins themselves.
Vesicle exocytosis is the fusion of a vesicle with the plasma membrane to release vesicular contents to exert various functions. This process relies on the cytoskeleton to transport the vesicles to the plasma membranes. There are three different modes of exocytosis, with them being the kiss and run, full collapse fusion, and compound exocytosis. In full collapse, the vesicle membrane fully fuses with the cell membrane. This process requires energy to separate and fuse the lipid membranes. This form of fusion creates a fusion pore which allows the content of the vesicle to be expelled. In kiss and run fusion, the vesicle temporarily fuses with the cell membrane long enough to create a fusion pore. The vesicle then pulls away from the cell membrane and reforms before returning to the interior of the cell. For compound fusion, secretory vesicles interact with each other during fusion. Compound exocytosis may occur sequentially with an initial primary fusion event at plasma membranes. results in a stable fused vesicle which becomes a target for secondary fusion events with deeper lying vesicles. Karp-1-coated vesicles are responsible for carrying misfolded proteins from the endoplasmic reticulum Golgi intermediate complex and Golgi back to the ER in a retrograde direction. There are two different types of secretion. There is regulated and constitutive secretion. In constitutive secretion, materials are transported in secretory vesicles to the extracellular space from the sites of synthesis in a continuous manner. Most cell types have at least one type of secretory proteins that are secreted in this manner. This form of secretion contributes to the formation of the extracellular matrix and plasma membrane. This secretion occurs through tubular transport carriers that bud out from the trans-Golgi network. Upon reaching the cell membrane, the secretory vesicles merge with the plasma membrane of the cell via exocytosis. The content in the vesicles are released into the extracellular matrix, and the lipids and membrane of the vesicles are donated to the plasma membrane. In regulated secretion, the materials are stored within a regulated secretory vesicle or granule until an external stimulation by a hormone or neurotransmitter causes it to fuse with the cell surface. Materials that are discharged are normally proteins needed intermittently, such as polypeptide hormones and digestive enzymes. These proteins lack a universal sorting system but instead involve physical sorting, selective retention, and condensation of secretory proteins through charge neutralization, protein aggregation, and active extrusion of ions. A way to regulate secretion would be through the action of proteins such as phenyltoctamin, which is the calcium sensor involved in the calcium 2+ ion-dependent exocytosis of synaptic vessels. Another method would be to control the protein-protein interactions of components of the fusion apparatus such as SNAR proteins. The SNAR hypothesis for membrane fusion states that some SNAR proteins act in trans. Due to this, a V-SNAR on the vesicle must interact with a T-SNAR on the plasma membrane to form a core complex which then mediates fusion. These proteins need to be targeted to specific cellular destinations. This is done by virtue of sorting signals that are encoded in the protein's amino acid sequence or is attached in the oligosaccharide. The receptors in membrane or surface coats of the budding vesicles recognize the sorting signal. Thank you for watching our video. If you enjoyed the video, leave a like and subscribe, and maybe leave a comment down below.